Hello everyone, and thank you for coming to my presentation. My name is Brooke Dominski, and I will be talking to you about the effects of acute ethanol exposure on learning in zebrafish. Zebrafish have been known to be a model in neuroscience when studying learning. They share approximately 76% of the genes that humans do. We use them because they are inexpensive and can be easily housed. One thing that makes our study unique to previous research is our large sample size. Many other studies use only about 20 fish when doing these studies. However, we used 188 for our study. With that, we can make more accurate conclusions while adding to the previous research that has been done. Before I get into this study, I just want to define a few terms for you all so you can have a better gauge of this study. The first is associated learning, which is what we were looking for in the fish. This is a form of conditioning that states behavior can be modified or learned by a stimulus and a response. So in this study specifically, the stimulus is the shock and the response is if the fish traveled back to that side of the tank after the testing period. The next keyword here is ethanol. We use ethanol in this experiment by putting it in the water. It's essentially the same found in human alcoholic drinks and is not harmful in low doses to animals. Essentially, they breathe it in and after a while it is out of their system, just like when humans drink alcoholic beverages. So our hypothesis here was that fish exposed to greater concentrations of ethanol would actually have more difficulties learning the conditioned behavior reflected in patterns in freezing, triggering shock, and speed of locomotion compared to those with lower doses of alcohol. With all of that background information now, in order to do this procedure, I would randomly assign each fish to their selected dosage, which were no dosage, low dosage, and high dose of alcohol, and randomly selected for which side of the tank the shock would come from, either the right or the left. Next, I would put each fish into their experimental tank, as you can see in this picture. The experiment then ran for about 90 minutes, and this same procedure continued for each fish within each of the 5 boxes that we had. Within the experiment, there were 4 different phases, as you can see on this timeline. The first was 10 minutes of acclimation so the fish could settle into their new environment, 10 minutes of baseline, 10 minutes of training, this would be the shocking period, and 60 minutes of testing. However, in our data, which will be coming up on the next few slides, we will only be comparing the testing and training period to see if the fish would associate the shock with essentially punishment and then learn to not go back to that side of the tank. After each experiment was done, we could then look at their individual heat maps as shown down here on your slide. So this is done by using infrared light to track the fish during the experiment. From there, we would use this to convert the data into quantifiable data, as you can see in the upcoming slides from the graphs. From our heat maps, we could use the new quantifiable data to compare phase 3 and phase 4, and by doing so, we could see if the fish actually altered their behavior, such as avoiding the side of the tank after the shock phase. Freezing was when a fish would stop swimming at any point in time. From there, we would compare phase 3 and 4, as I've said before, and found actually no significant differences in freezing compared to alcohol doses, thus concluding that freezing was not significantly affected by dosage of ethanol. Triggers then were defined as how many times the fish was shocked during the testing period. During the testing period, we saw significant differences between the doses, with high dose alcohol having the most triggers while no dose alcohol having the least amount of triggers. And then we have speed. So speed in this case was an average of how fast the fish swam during that phase. With that, as you can see here, the higher dose alcohol conditions resulted in higher average speed than no dose alcohol. So in this study, we examined the effects of alcohol on behavior during testing and training and found that triggering and average speed were associated with alcohol doses, which provides evidence for our hypothesis that greater concentrations of alcohol will decrease learning, while lower doses of alcohol actually do not. However, we did not see any evidence that alcohol affects freezing behaviors. So in summary, our evidence supports previous studies that zebrafish show evidence that higher doses of alcohol can decrease learning, while lower doses of alcohols did not decrease their learning. It also supports the evidence seen in prior studies done with the smaller samples of zebrafish. Lastly, I would just like to thank Dr. Sheese, who is my dependent study professor, and the IWU Psychology Department for helping us through this whole process. Thank you.